3 new therapies just showed incredible promise for ApoE4 brains. One targets 4 proteins at once and another tightens the blood-brain barrier. And the third one is particularly surprising. Welcome back, I'm Dr. Kevin Tran, and in this video we're breaking down the most important findings from the International Conference on Alzheimer's and Parkinson's disease and related neurological disorders that happened in April 2025. So this is extremely fresh research. This was a dense data-rich event, but I've distilled the most critical insights for you as an ApoE4 carrier. We will cover why ApoE4 disrupts your brain on a cellular level, how rare protective variants like ApoE2 or Christchurch are reshaping our treatment roadmap, new therapies in clinical trials that may finally work for E4 carriers and not against us, and fourth, what all of this means for the lifestyle, labs, and protocols we're already experimenting with. So whether you are E3, E4, 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 or supporting someone who is, this video is your cheat sheet to what's coming next in prevention, intervention, and hope. Let's get into it. Let's start with a question that haunts every ApoE4 carrier. Why does this gene variant raises our risk for Alzheimer's so dramatically? It's not some random curse, it's the legacy of a powerful but now outdated adaptation. ApoE4 isn't just a bad gene, it's an ancestral survival tool caught in modern mismatch. And at the center of it is the fat metabolism. How your brain moves lipids around is either your secret weapon or your silent saboteur. Let's break down how ApoE4 affects your lipid biology, why it used to be helpful, and what this means for your health today, especially if you're carrying these copies of ApoE4. Let's rewind all the way to the gene itself. ApoE, short for apolipoprotein E, is a gene that helps shuttle fats, including cholesterol, around the body. But here's the twist. Humans are polymorphic for this gene. That means we have 3 major variants, ApoE2, ApoE3, and ApoE4. Most mammals only have one version, actually. We have 3. And the difference between them, just 1 or 2 amino acids. By the way, this makes it for a perfect target for CRISPR, for gene editing. But coming back to these tiny changes, these 1 or 2 amino acids have massive ripple effects across the brain. Think of these proteins like differently shaped keys. ApoE2 might fit the lock loosely. ApoE3 fits just right. And ApoE4, it forces the lock open but leaves damage behind. Let's hear how Dr. Henrietta Nielsen framed this unique human trait.
Other mammals that we also normally study in the lab have just one ApoE, they're monomorphic. And as you can see in this table, the 3 major isoforms in humans are the E2, E3, and E4.
So what makes E4 different? ApoE proteins are like delivery trucks for fat. They carry cholesterol and lipids through the bloodstream to where they're needed, especially in the brain, which is 60% fat by dry weight. But ApoE4 is like this truck with the wrong GPS. It delivers the cargo to the wrong place or crashes into the gates. These crashes create traffic jams in your brain lipid system, and those jams are tied to inflammation, beta amyloid buildup, and blood barrier dysfunction. E4 carriers often absorb or retain more dietary fat in the gut because thousands of years ago, this was an advantage. You wanted to hold onto every calorie and molecule of fat. Food was scarce, survival was the goal. And by the way, the fact that we absorb more dietary fat means that molecules like ezetimibe works really well for us. Okay, let's hear how Dr. Henrietta explains this evolutionary mismatch.
And because of its role implicated in the modulation of lipid levels, ApoE4 is also a major risk factor for coronary heart disease. It's been proposed that ApoE4 increases the lipid uptake in the intestines, which in fact used to be beneficial since it's the ancestral variant.
Here's the irony. What once helped us survive, basically maximizing fat absorption, is now hurting us in the world of processed food and sedentary lifestyle. ApoE4 carriers tend to have higher LDL-C, remnant cholesterol, and more oxidized lipids floating all around in your bloodstream. That's not just a cardiovascular risk, it's a cognitive one as well. These lipid particles activate inflammatory microglia, damage the blood-brain barrier, and impair cholesterol recycling in neurons, which then in turn affects synaptic plasticity, which is basically your brain's ability to learn and adapt. It's like trying to remodel your kitchen while the plumbing is leaking and the delivery guy keeps dropping off the wrong parts. But here's a key point. Not all ApoE4 carriers are equally at risk. Dr. Henrietta showed population-level data revealing major differences across ethnic groups. For example, Nigerians with E4 don't see the same Alzheimer's spike as Koreans or Europeans. That means other genetic factors or lifestyle, gut microbiome, infection, early nutrition, all of these factors are either buffering or amplifying the effect of E4. Let's look at the data she shared.
The ApoE4 promoter risk of Alzheimer's disease appears to vary across populations. For example, in some African populations where we have the odds ratios here for E3, E4 subjects and for homozygous individuals, where you can see that the risk, for example, of Alzheimer's disease in the Nigerian population is not at all as high than some of the other populations, like for example in the Korean population where you might have a 25-fold higher risk of Alzheimer's disease if you are homozygous.
What does that mean for us ApoE4 carriers? Let's translate this science into action. One, your lipid panel isn't optional. This is essential to do regularly. You need to know your ApoB, your LDL particle count, your triglycerides, your HDL, and your oxidized LDL. These are not just cholesterol numbers. They are early warning signs for how your brain handles inflammation and energy transport. Second, the ancestral mismatch is real but reversible. ApoE4 isn't broken, it's just outdated. So your diet should reflect that. Diet lower in refined carbs, rich in omega-3s that you find in fish, for example, and mindful of saturated fat tend to work better for ApoE4 carriers. Third, your ancestry and environment still matter. Whether you're African, Asian, or European, ApoE4 doesn't act in a vacuum. Your microbiome, childhood infections, and lifelong inflammation all modulate how this gene expresses itself. So, if you are carrying ApoE4, this is your call to get curious, not panicked. Your genetics are just the start. What you do with this information, that's what defines your risk and your future. Moving on to the second insight in this video. When we talk about ApoE4 and Alzheimer's, we almost always picture the brain, like neurons, plaques, tangles. But what if one of the most powerful drivers of that risk wasn't even in the brain? Can you guess which organ it is in? It's in the liver. And understanding that could change everything, from what you track in your labs to the lifestyle changes that could actually move the needle for you. Because for ApoE4 carriers, your brain and your liver are locked in a long-distance conversation. Let's unpack what that means and why ignoring your liver could mean missing half of the ApoE4 story. Okay, let's start with a quick refresher. ApoE is produced both in the brain and in the liver, but the liver is the main source of circulating ApoE, the version floating around in your blood, called peripheral ApoE. And here's the conventional wisdom. That peripheral ApoE doesn't cross into the brain because it's blocked by the blood-brain barrier, or BBB. Think of the BBB like an elite border checkpoint. Only selected molecules can get through, and ApoE from the liver isn't on the guest list.
Importantly, peripheral ApoE does not cross the blood-brain barrier, and that's been shown in several liver transplantation studies.
So, if liver-produced ApoE4 isn't sneaking into the brain, how can it still influence Alzheimer's risk? Well, here's the key: indirect effects. Your liver sets the tone for your whole body metabolic environment. It regulates lipid levels, inflammation, energy balance, and all of that affects your brain. Think of the liver as the climate control system. If it's generating chronic metabolic storms like high LDL, oxidized fat, or low-grade inflammation, that weather pattern reaches the brain whether the ApoE molecule crosses the barrier or not. This is why liver health might be one of the most underappreciated tools in the ApoE4 playbook. And here's where it gets really actionable. Researchers in Denmark looked at over 100,000 people. They found something striking. People with low levels of plasma ApoE had significantly higher risk of not just Alzheimer's, but all-cause dementia. That means the amount of ApoE in your blood may be just as important as the type.
It's the low plasma ApoE levels that seem to be linked to not just an elevated risk of Alzheimer's disease, but all dementia. They looked at some 100,000 individuals, and they show that the individuals with the lowest level of plasma ApoE are also the ones with the highest odds ratios for not only Alzheimer's disease, but all dementia.
To really unpack this connection between liver and brain, researchers built what were called humanized liver mice. Here's how it works. They take mice and transplant them with human liver cells called hepatocytes. These cells make ApoE, just like basically in us humans, and this lets researchers study how human E4 livers behave in a living system. And the results are eye-opening. When these mice are populated with E4 hepatocytes, their entire lipid profile shifts into a more human pattern with high LDL, altered HDL, and other plasma protein changes. It's like flipping a genetic switch that alters the entire systemic metabolism.
The majority of the lipids in the mice are in the HDL particles, whereas human, the opposite, we have most of our lipids in LDL particles. So if we use this mouse model, repopulate them with human primary hepatocytes, we can shift this LDL, HDL to a very human-like profile. So not just the lipids turn human-like, but also other plasma proteins, and that's what we really like about this model.
Now, here's the big provocative question. Is it possible that just having ApoE4 in the liver, not the brain, might be enough to drive the E4 disease risk? Dr. Henrietta left us with that exact speculation. If that's true, it will completely redefine our approach. It will mean that liver-targeted therapies, whether dietary, pharmacological, or even gene-based, could be leveraged to reduce risk without ever crossing the blood-brain barrier.
So we speculate whether the liver ApoE genotype might be sufficient to produce a potential ApoE4 phenotype.
What does it mean for us, ApoE4 carrier? Well, one, your liver is not just a background character. It's actually a lead actor. If you're not actively tracking liver health, lipid ratios, and inflammatory signals, you're only seeing half the ApoE4 picture. Second, low plasma ApoE equals high dementia risk. Third, support your hepatocytes, whether it's reducing or completely eliminating alcohol, intermittent fasting, omega-3s, or managing insulin resistance. All of these interventions will increase your liver efficiency and be completely brain protective by proxy. And fourth, we may not need to cross the blood-brain barrier to change the game. If liver-based ApoE4 alone is enough to shape the risk phenotype, then therapies that never touch the brain might still profoundly lower the risk. That includes metabolic optimization, inflammation control, and even gut-liver-brain axis intervention. So overall, your liver is doing more than what you think. If you're an ApoE4 carrier, ignoring it could mean missing massive piece of the prevention puzzle. We're entering an era where brain health isn't just about neurons, it's about the entire body networks. And the liver is one of the most influential nodes in that network. Cool, quick check-in here. If this deep dive into liver biology gives you an aha moment about your own health, hit that subscribe button. That is exactly the kind of nuanced science-backed insight we're unpacking every week at the Phoenix and on this channel. If you're already experimenting with ways to support your liver and lipid profile, drop a comment below. I'd love to hear what's working for you. In this next segment, we're going to meet the ApoE variants that protect against Alzheimer's, like ApoE2, Jacksonville, and Christchurch. By studying these outliers, these biological success stories, scientists are reverse engineering resilience. And for ApoE4 carriers like us, that means hope. Because the more we understand about why these variants protect the brain, the more we can mimic those benefits, even if we didn't win the genetic lottery. So let's start with the ApoE Jacksonville variant, a rare mutation discovered in a patient from, well, you guessed it, Jacksonville in Florida in the US. What makes it special? This variant reduces ApoE self-aggregation. Normally, ApoE4 tends to clump together, forming something we call tetramers, which are clusters of 4 proteins, tetra4. That clumping makes it very sticky, dysfunctional, and potentially toxic, basically like amyloid or tangles. You don't want to have these clusters. So the Jacksonville variant resists this clumping. It stays as a monomer, basically a single smooth functioning unit, and that means better cholesterol transport, less gunk in the system, and a healthier lipid environment in the brain. Let's hear about Dr. Bu explain this exact mechanism.
We described the ApoE Jacksonville variant. This was discovered when I was in Mayo Clinic, Jacksonville, Florida. And the mechanism we found is reducing ApoE self-aggregation. So normally when you express ApoE in, for example, HK cells, you get these tetramers. So that's very stable conformation for ApoE. This is the ApoE3 backbone. But if you have the Jacksonville variant, this become monomer. It actually is a better cholesterol acceptor.
Now let's talk about ApoE2, the most well-known protective variant. Dr. Bu's team studied mice with different ApoE genotypes. They looked at 2 places, cholesterol inside brain tissue and cholesterol circulating in the cerebrospinal fluid, or CSF, that clear liquid that bathes your brain and spinal cord. What they found is fascinating. ApoE2 brains had less cholesterol accumulation in the tissue, what they call a lean brain, but at the same time, ApoE2 particles in the CSF were carrying more cholesterol. That suggests ApoE2 is doing a better job moving cholesterol through the brain rather than letting it build up inside it. That is crucial for synapses. Those delicate junctions where brain cells communicate Cholesterol is vital for synapse formation, but too much in the wrong place causes inflammation and dysfunction.
We found that E2 brain, at least in the green matter, has reduced level of cholesterol. We call this lean brain. And this reduced cholesterol correlated with better memory performance. If you look at the CSF particle, ApoE particle is the opposite. So E2 associated with greater amount of cholesterol, and this is positively correlated with memory performance.
ApoE2 doesn't just move cholesterol better. It may change how brain cells function. Using single-cell RNA sequencing, Dr. Boo's team found that E2 upregulates genes involved in synaptic function and cholesterol biosynthesis. That means the E2 brain isn't just leaner, it's more metabolically tuned for maintenance, plasticity, and resilience. Let's look at what he shared.
Most of the genes or pathways are upregulated in E2. So this is excitatory inhibitory neuron. And if you look at the specific pathway, many of these related to synaptic function and also cholesterol biosynthesis.
Now, here's where things get personal and extraordinary. Imagine carrying one of the most aggressive Alzheimer's mutation known, PSEN1 or presenilin-1. Most carriers develop dementia in their 40s, but this woman didn't. She actually lived into 70s without any symptoms. Her brain had a high amyloid load, but her tau tangles were low, and her cognitive function remained largely intact. Why? Well, she was homozygous for the ApoE3 Christchurch variant, a rare mutation previously linked to familial hypercholesterolemia, but now emerging as a powerful Alzheimer's shield. Let's hear Dr. Shona Yuan explain her case.
This patient on the right, Illyria, she was able to defy Alzheimer's disease. She is a carrier of presenilin-1 E288 HISA mutation. The typical age of onset for this kindred is in the age 40s. However, she did not develop Alzheimer's disease at age 40. She was found to have mild cognitive impairment in her 70s. So how did that happen? She was shown to actually have higher level of amyloid. However, she was found to actually have lower level of tau. She was found to be a carrier and homozygous for a Christchurch mutation.
To figure out how Christchurch works, researchers created genetically identical brain cells in the lab known as isogenic iPSC-derived organoids. In these models, the Christchurch variant normalized a key Alzheimer's biomarker, the Abeta42 and Abeta40 ratio. That's huge because a high 42-40 ratio is a hallmark for Alzheimer's disease.
In the presenilin-1 line, there was elevated 42-40 ratio compared to the reverse presenilin-1, which has the presenilin-1 mutation corrected. The presenilin-1 mutation, which has a heterozygous or homozygous Christchurch mutation, where it behaving more like the reverse presenilin-1 line, suggesting that the Christchurch mutation may have a direct effect.
Here's why this matters so much for us ApoE4 carriers. One, it's proof that risk can be offset. These stories show that even with high-risk genes or even massive amyloid loads, you can resist the disease. The key lies in downstream effect, cholesterol transport, tau spread, protein aggregation. Second, we can learn from nature's playbook. ApoE2, Jacksonville, and Christchurch aren't just lucky accidents. They point towards specific mechanisms we can aim to replicate, even if we carry ApoE4. Drugs that reduce ApoE aggregation, support monomer formation, or shift lipid handling are already in development today. And third, your ApoE4 isn't your fate, it's a starting point. Whether it's through better synaptic function, reduced tau pathology, or enhanced cholesterol transport, these protective variants gives us target. In the Phoenix community, we are already running structured experiments to test which lifestyle interventions might move us in that direction. So don't see ApoE2 stories as not relevant and these guys are so lucky. See them as blueprints for brain resilience for us ApoE4. So far, we have explored ApoE4 at the genetic level, the liver level, and even in the rare protective variants. But now we go inside the cell. This is where ApoE4 becomes less of a theoretical risk factor and more of a microscopic saboteur. It corrupts the function of glial cells, basically your brain's frontline defenders, leading to failed garbage disposal, inflammation, and toxic buildup. Think of it like traffic jams on your brain's internal highways. The cleanup crews can't get through, the trash piles up, and eventually everything starts to break down. Let's look at how this chaos unfolds cell by cell. First up, astrocytes. These star-shaped glial cells do everything from feeding neurons to cleaning waste. In healthy brains, they help vacuum up amyloid beta, one of the toxic proteins linked to Alzheimer's. But ApoE4 astrocytes, well, they're lousy janitors. Dr. Ojakor's team used iPSC-derived astrocytes, essentially brain cells grown from stem cells, to test how well they could clean up amyloid. The ApoE3 cells took up significantly more amyloid than ApoE4.
If you treat iPSC-derived astrocytes expressing either human ApoE3 or ApoE4 with amyloid beta for 2 days, and you just look to see the amount of A beta that was taken up by these cells, and ApoE4 astrocytes take up significantly less A beta compared to ApoE3 astrocytes.
Why does this happen? Well, part of the answer lies in the structural protein called JFAP for gliofibrillary acidic protein. JFAP forms part of the astrocyte's internal skeleton, but it's more than just scaffolding. It helps guide endosomes, which are the tiny cargo containers carrying things like amyloid beta through the cells. In ApoE4 astrocytes, JFAP levels drop as the animals age. That means these cargo trains lose their track. Amyloid gets stuck, misdelivered, and left to rot inside the cell.
We found in these mice was that there was a steady and consistent decrease in GFAP expression in ApoE4 mice at 8 months. If you look at whole brain section from these mice, you can actually visibly see the loss of GFAP in these mice. We wanted to know if this was a progressive loss of GFAP or if this was a sort of a lifelong defect. And so we looked at neonatal mice at P3, so 3 days post-birth, and we just looked to see if GFAP levels were changed between E3 and E4 cortical samples. So that tells us that this loss of GFAP is actually related to the age of these mice.
So now, even when amyloid beta is taken up by the cell, it can't get where it needs to go, which is the lysosome, your cell's incinerator. These researchers showed us that with low GFAP, amyloid doesn't effectively reach these lysosomes. It just sits there clogging up the works. Imagine a garbage truck that picks up the waste but gets lost on the way to the landfill. The trash stays in the truck, rots, and causes more damage.
We saw that astrocytes with less GFAP actually held on to A beta for much longer than regular astrocytes expressing regular GFAP. But interestingly, we see that GFAP knockdown astrocytes actually are A beta localizes significantly less with lysosomes in these cells.
This brings us to the Rab protein, a family of molecular zip codes that helps vesicles, the vesicles are the cargo bubbles, find their destination. Rab5 directs early endosomes, Rab7 handles late endosomes, and GFAP appears to interact with both, helping sort and shuttle these containers. But in ApoE4 brains, when GFAP goes down, these interactions collapse. The system loses its internal GPS.
When you lose GFAP, the number of these GFAP Rab complexes is also diminished. In ApoE4 mouse brains at 8 months, you actually lose the interaction that occurs between GFAP and Rab5, as well as the interaction between GFAP and Rab7.
Now let's shift to microglia, the brain's immune cells. Normally, they patrol the brain, gobbling up debris and calming inflammation. But under ApoE4, they get sluggish, inflamed, and worse, they start to accumulate lipid droplets. These droplets are basically like blobs of fat inside the cell, and it clogs their metabolism and blocks their ability to clean up amyloid. Dr. Bu showed us that ApoE3 microglia could reduce amyloid buildup. ApoE4 microglia, not so much.
You can see that E2 give you the greatest activation, then E3, and then E2. If you look at the E4 microglia, not only they have reduced activation, but they also have lipid accumulation in the form of lipid droplet. And here's staying with plane 2. And this ApoE4 defect actually have multiple level, reduced activation, phagocytic function, and also reduced lipid efflux and accumulation as lipid droplet. If you look at breed this to amyloid pathology, we know that microglia E3 reduce the amount of amyloid. E4 does not have that effect.
But there's good news. Remember the Christchurch variant from the previous insight? Well, Dr. Yuan showed that this protective mutation can rescue the microglia, even in the context of severe Alzheimer's mutation. In her model, Christchurch reduced lipid droplets buildup, suggesting it restores microglial health and function.
We see that in the Presenilin one, there's a relatively higher level of lipid droplet formation compared to the reverse Presenilin one. This also can be rescued by the homozygous Christchurch mutation in the Presenilin one mutation line.
So, what does it mean for an ApoE4 carrier? Well, this is about so much more than just plaques. ApoE4 disrupts the cleaning crew, which are the astrocytes and the microglia, and turns your cellular highways into traffic jams. So interventions must go beyond targeting amyloid. We need to restore the function of the cells themselves. Glial cells health is brain health. In the Phoenix community, we look at interventions that support astrocytes and microglial function, like lifestyle changes that reduce chronic inflammation, improve autophagy, and promote lysosomal health. Number 3, biomarkers can and should guide us. We track more than just cognition. We look at metabolic inflammation, lipid profiles, and mitochondrial function as proxy for how your glial cells might be faring. And 4, everything is fixable or at least modifiable. Whether it's through nutrition, supplements, or future therapies, the goal is to nudge ApoE4 brains towards the more resilient E3 or E2 phenotypes. And that starts by keeping your cellular cleanup crews online. Cool. Now let's talk about solutions. So far, we've explored the cellular damage ApoE4 causes and how it derails lipid transport, immune function, and protein clearance. But here's the exciting part. Scientists aren't just observing the damage, they're designing ways to intervene, some of which are already in human trials. At this year's conference, we saw therapies that 1, reduces multiple toxic proteins at once, 2, repair the blood-brain barrier, 3, reprogram glial behavior, and 4, even switch out ApoE4 for ApoE2 at the genetic level. This is where science becomes hope. And for ApoE4 carriers like us, it's no longer about waiting for a miracle. It's about tracking which frontier breakthroughs we are most likely to benefit from. First up, buntanetap. This compound doesn't pick just one target. It goes after 4 of the most toxic proteins involved in Alzheimer's: the amyloid precursor protein, tau, alpha-synuclein, and TDP-43. And it does this by targeting a regulatory region on the 5 untranslated region, so 5 UTR, of the mRNA, a kind of master switch that controls how much of each protein gets made. Let's hear Dr. Cheng Fang walk us through the mechanism.
Our lead compound is called buntanetap. It inhibits the production of multiple neurotoxic aggregating proteins simultaneously because the 5' untranslated region of the mRNAs of APP, alpha-synuclein, tau, and TDP-43, they all have this conserved atypical IRE, the little loop here.
In their phase 2 and 3 trial, buntanetap worked best in people with mild Alzheimer's, and critically, it seemed to benefit ApoE4 carriers just as much. That's very rare, because many antibody drugs either don't work as well for E4s or come with a higher risk of brain swelling. Even more impressive, buntanetap reduced levels of NFL, or neurofilament light chain, which is a key marker of ongoing neuronal damage.
In mild Alzheimer's disease, you can see a very clear dose-dependent treatment effects, and especially in 13, 15, and 30 mg, nominal clinical statistical significant improvement, both to the baseline and also to the placebo. If you look at the genetic components, whether they are ApoE4 carriers or not, they still have treatment effects. This is very important because we know that in current monoclonal antibody treatments, ApoE4 carriers don't have the same effects, and also they're prone to ARIA. So is NFL, which is axonal marker used to show that axonal integrity and neuronal health. So this is quite remarkable. We saw that in placebo group increased, which shows there more neuronal damage. And in treatment groups actually reduce NFL, showing that we are preserving more axons and more neuronal functions.
This is a PDE5 inhibitor. If this rings a bell, it's because it's the same class as drugs for erectile dysfunction. But in the brain, it works very differently. Basically, ApoE4 carriers have weaker blood-brain barriers. We've already seen that at the beginning of this video, due in part to lower levels of Claudin-5, a protein that zips brain endothelial cells together. When Claudin-5 drops, your brain becomes more permeable, allowing toxins, pathogens, and inflammation to sneak inside the brain. Myrodenafil helps reverse that. In E4 mice, it boosted blood flow, restored Claudin-5, reduced vascular amyloid, calmed the microglial inflammation, and even improved memory. Let's break down the data.
Claudin-5, or CLN5, and it's a critical tight junction protein in the blood-brain barrier. In ApoE4 carriers, there's a reduction in CLN5 expression. Because there's reduced expression of this CLN5 in ApoE4 carriers, it weakens the blood-brain barrier integrity, it increases permeability, and therefore allows harmful substances to enter the brain. On the top right, that the FITC see dextran perfusion is significantly improved after miradenafil treatment, indicating enhanced cerebral vascular flow. Panel B shows immunostaining for the CLN5 and the CD31 in hippocampal blood vessels. Compared to vehicle-treated ApoE4 mice, miradenafil-treated mice exhibit increased CLN5 expression, suggesting improved tight junction integrity and blood-brain barrier function. In panel C on the bottom left, we examine A-beta deposition. Vehicle-treated mice show strong A-beta 42 accumulation around blood vessels and in the CA3 region, and after treatment with miradenafil, there's reduction in the vascular A-beta, again demonstrating its role in reducing amyloid burden around the compromised vasculature. This both suggests a shift towards an anti-inflammatory phenotype. Next, we move to an in vitro model using LPS-stimulated BB2 microglial cells, and miradenafil dose-dependently suppressed pro-inflammatory cytokines, confirming a direct anti-inflammatory effect on the microglia. The Y-maze test showed in panel A, the knock-in mice exhibited reduced spontaneous alternation, which indicates impaired spatial working memory, and the miradenafil-treated group significantly improved their performance, suggesting enhanced short-term memory.
Now this one is mind-blowing. What if we could switch your ApoE4 gene to a protective ApoE2, but only in specific brain cells and only when needed? That's exactly what Dr. Leslie Gordon's lab did using the CRELOX system, a genetic toolkit that lets scientists perform precise tissue-specific edits in living animals. In a model of Alzheimer's, which is the 5X FAD, they flipped the switch from E4 to E2 only in astrocytes and only at late-stage disease. The results? Well, less amyloid, reduced glial inflammation, and improved memory performance.
We developed what we call the ApoE switch mouse, which I'll refer to as 4S2 for the rest of this presentation. And this mouse model uses the CRELOX-P system to allow for an inducible and cell type specific transition from E4 to E2. It's a floxed mouse model, and at baseline, this mouse is only going to express ApoE4. When we used our associative fear conditioning, was that this late stage switch from E4 to E2 in only our astrocytes was enough to not only significantly improve associative learning, but it improved their contextual and cued memory when compared to the mice who did not switch. When we look at animals who are now expressing astrocyte-derived ApoE2, we see that there is not a significant increase in that 2-month time period, suggesting that while we're slowing deposition rather than improving clearance, we see that there is a significant reduction in both microglial and astrocytic reactivity when we switch to expression of astrocytic E2.
But here's the twist. When they made the same genetic switch but only in microglia, amyloid still went down, but memory didn't improve. In fact, microglial inflammation went up. This shows that context matters. It's not just the gene, it's which cells expresses that gene, and when they express it, and how they interact with the environment.
Interestingly enough, following the same experimental paradigm, we did not see an improvement in either associative learning nor contextual or cued memory. So there's something going on there. However, like we see with an astrocyte-specific switch, we are seeing a significant reduction in total amyloid levels. When we switch specifically microglia from E4 to E2, we're not seeing any differences in astrocyte reactivity. However, we're seeing a significant increase in microglial reactivity.
This segment gives us a powerful new framework and a lot of insights. One, the future is definitely multi-targeted. Complex diseases require complex solutions. Drugs like buntanetap that targets multiple proteins at once may offer a better path forward for ApoE4s than overly narrow approaches. 2, vascular health is brain health. Miradenafil reminds us that strengthening the blood-brain barrier through drugs or lifestyle can reduce inflammation, improve cognition, slow disease. 3, precision is coming even though complexity remains there. Gene switching is powerful, but it's also a reminder of how sensitive the brain ecosystem is. The same intervention in a different cell type can lead to opposite results. For now, this underscores the importance of supporting every cell type with a systemic whole-body strategy. And 4, the science informs what we do today. While these drugs are still in development, their mechanism validates what many of us are already experimenting with. Basically, reducing inflammation, supporting lipid and cholesterol metabolism, enhancing blood flow, and tracking neuronal damage via biomarkers like NFL, GFAP, and HSCRP. And this is basically the essence of what we do inside the Phoenix community. We run structured, personalized experiments that mirror where the science is going, not where it used to be decades ago. Before we wrap up, let's quickly recap what we covered in today's video, because each of these insights gives you more leverage in your fight against Alzheimer's. Insight number one is about how ApoE4 is an ancient gene that helped our ancestors survive, but in today's food environment, it disrupts lipid metabolism and raises Alzheimer's risk. Insight number two is that the liver plays a much bigger role than what we thought. Even though liver-made ApoE doesn't enter the brain, it shapes the body-wide inflammation and lipid environment that impacts brain health indirectly. Insight number three is how protective ApoE variants like ApoE2, Jacksonville, and Christchurch offer a blueprint for resilience, and scientists today are learning how to mimic their benefits. Insight number four is showing how ApoE4 sabotages brain cells at a microscopic level, disrupting cleanup crews like astrocytes and microglia, and cause traffic jam inside cells that lead to toxic buildup. We have a whole other video on astrocytes and microglia because these deserve a deep dive. You can look it up in our channel to not miss it. Insight number five is how new therapies are on the horizon. From multi-target drugs like buntanetap to BBB repair strategies like mirodenafil, and even gene switches that convert ApoE4 to ApoE2 in specific brain cells. The takeaway is that all of these are not just academic findings. These are stepping stones towards a new model of prevention, one where your genetics are just one input and your choices, your life choices, carry the rest. In the Phoenix community, we translate this science into action. We track biomarkers, we test interventions, we help each other figure out what works through structured experiments, expert Q&As, and real-time support from people who share this genetic reality. We're not waiting for a cure, we're building resilience today, right now. If you are an ApoE4 carrier and want to beat the odds and defeat Alzheimer's, check out the Phoenix community. The link to apply is in the description. I review every application personally to make sure it's a great fit. So if you're coming from YouTube, just mention that in your note and I'll make sure to show you some extra love. All right, that's it for today. I wish you a great day and see you in the next video.